The subculturing of mammalian cells is an important step for the maintenance of the cells as well as preparation of cells for cell culture experiments. To do this, there's a number of different cell types. These include both suspension cells and adherent cells. We're using very common cell lines, including the human embryonic kidney cell line, epithelial cell lines, including the HT1080 cell lines, and these use very common media. This media composes of the MEM growth media, which contains fetal bovine serum and antibiotic. All of our reagents and materials must all be sterile before use, and we take great care in cleaning of our work surfaces as well as our gloves while working in cell culture incubator. To passage cells, the first step is to collect the cell culture media. We'll need the trypsin, MEM, and the PBS. The cells are incubated in a hot water bath set at 37 degrees Celsius. They're incubated for 10 minutes. I will start by preparing the cell culture incubator. We'll turn on the lights and the fan and lift the sash. And then the next step is to wipe down the interior with 70% ethanol. This is a paper towel I'm using to wipe down the interior. I'm giving special attention to specific items, including pipette men and tips that I plan to use for the cell culture work. I am transferring the trypsin as well as a glass beaker for waste collection into the hood and spraying down with 70% alcohol. While the cell culture media is incubating, I'll examine the confluency and the health of the cells. So I'll remove the cells from the 37-degree incubator and examine these with the microscope. I will adjust the focus and examine the confluency. These look to be roughly 40% confluent. I can also use the camera to image and record the confluency for my records. I'll just adjust the focus for improve the quality of the image. Now I can turn the microscope off and remove the cells for subculturing. Now that the media has reached 37 degrees Celsius, we can remove the media from the hot water bath and wipe down the excess water on the outside of the media containers. And we will then wipe these down with ethanol as we transfer them into the cell culture plate. Let's freeze that one more time to remove the excess ethanol from the surface. The next step is to aspirate off the cell culture media from the plate. So I'll use the vacuum line and a sterile serological glass pipette to aspirate off the media from the cell culture plate. The next step is to wash the cells with 10 milliliters of the PBS wash buffer. So I'll use a sterile serological pipette and my pipette aid to draw up 10 mils of PBS. I will gently transfer the 10 mils of PBS to the cell culture plate, avoiding directly placing the PBS on the cells to avoid dislodging the cells from the surface of the plate. This step is to wash the remaining cell culture media from the plate. I will just gently mix to ensure that remaining cell culture media has been mixed well with the PBS. The next step is to aspirate away the PBS buffer. Again, I'll use the vacuum and a fresh sterile glass pipette to aspirate away the PBS wash buffer. The next step is to transfer 1 milliliter of the trypsin enzyme to dissociate the cells from one another and from the surface of the plate. So I'll use a sterile pipette to transfer the trypsin to the plate. I'll do this in a dropwise fashion across the entire surface of the plate. And then I'll just spread the entire contents of the trypsin across the entire surface of the plate by gently rocking the plate to ensure that the trypsin is covered the entire surface. Then I will transfer the plate to the 37-degree incubator to allow for the trypsin enzyme to work. The adherent cells that we use have a very common media, so we're working with an adherent cell line, the human embryonic kidney 293T cells, but the cell media, the DMEM, Dubocal's modified eagle medium, is a common media. To prepare this media, we also need to add fetal bovine serum and antibiotic. So I'll first add the fetal bovine serum, so the total volume that I will add is 50 milliliters. So I'll use a sterile serological pipette to transfer the fetal bovine serum to the DMEM media. I will next add 5 milliliters of the antibiotic to the fresh DMEM media. This prevents bacteria from growing inside of our cell culture. Then I mix the media well before use. I will also label the media with my initials and the date, as well as the contents. Now that the 5-minute incubation is complete, I'll remove the cells from the cell culture incubator. And transfer them back to the cell culture hood. I'll transfer 10 milliliters of the fresh DMEM media to the cell culture plate using fresh, serological pipette. I'll then resuspend the cells. So I'll draw up the media and then work on washing the remaining cells from the surface of the plate. After resuspension, I'll transfer these cells to a fresh 15 ml conical tube. The plate and any biological waste can be transferred to the biological waste container, and this will then be collected for autoclaving. To wash the cells and remove the dead cells, I will pellet them using a centrifuge. The cells will be spun at 300 times G at room temperature for 5 minutes. At end of run, I can remove the cells from the centrifuge. And the cells should now be pelleted. And these will then be transferred back to the cell culture hood for aspiration of the supernatant. The next step is to use the vacuum to aspirate away the supernatant. And then the next step is to resuspend the pellet and 10 mils of DMEM cell culture media. So I want to resuspend the cell pellet completely and the fresh media. To determine the total number of cells and the percent viability, I'll take the slide and the trypan blue membrane exclusion dye to mix with our cell sample. I'll take one of our sterile 1.7 mil Eppendorf tubes. Or collect 50 milliliters of the trypan blue exclusion dye. Transfer that to our fresh Eppendorf tube. Collect 50 milliliters of our resuspended cells. And transfer this to our trypan blue dye and mix thoroughly. Then I'll take 20 microliters of the freshly prepared cells and transfer these to the slide, allowing for diffusion to transfer the cells across the surface of the slide. The slide can now be taken to our cell counter. To count the number of cells and the viability of cells, I'll insert the slide into the cell counter. The counter will focus on the slide and perform the cell counting. I can record the number of cells as well as the percent live cells in my sample. The next step is to collect a fresh sterile plate to transfer the cells. I will take a marker and label the fresh plate with the name of the cell line. My initials, today's date, and then I will transfer the resuspended cells to the fresh plate using a new serological pipette. For the 15 centimeter diameter plate, the total volume of media is 20 mils. So I need to bring up the remainder of the volume up to 20 mils with fresh DMEM cell culture media. So So again, I'll use a serological pipette to transfer the remaining volume. I want to avoid having any air bubbles. The final step here is to gently spread the cells across the entire surface of the plate by gently rocking the plate back and forth. The plate is now ready for incubation in Subculture incubator. After preparation of the fresh plate, the cells can now be transferred to the cell culture incubator. To clean up the cell culture incubator, I will cap our serological glass pipette tips, turn off the vacuum, reconnect our serological pipette aid back to the charger. The additional waste, the serological pipettes and 15 ml conical tube can be discarded. And I use 70% ethanol to spray down the contents of the cell culture hood. Particularly clean all the surfaces in which I worked.